All right, so this is chapter 2 for human anatomy, the cell. We're starting on Roman numeral 2 in the outline, the eukaryotic cell. So this part is going to concentrate on the membrane, and then part 3 is going to concentrate on the organelles. So here we have a picture of a eukaryotic cell. So letter A says, well, what is a eukaryotic cell? What makes a cell eukaryotic versus the other kind of cell, prokaryotic? So eukaryotic cells, number one, it contains internal cell membranes. So if you see these organelles here, these discrete units, I'm going to circle the mitochondria. So that's an organelle. This organelle is created by membranes, so it encloses the interior of the mitochondria, and that makes sort of a very specific area that can be different than the surrounding cytoplasm. So the cytoplasm here, the X's, that could have chemicals, it could have different chemical reactions and different molecules in it than that's inside the mitochondria. So we can create these distinct microenvironments, different environments within the cell. So that's what number 2 says. Internal membranes create microenvironments. That means that there's a division of labor. So what goes on in the mitochondria is not going to be the same as what goes on in, let's say, the Golgi apparatus. So it's a higher level of complexity for eukaryotic cells. So, and they're also much bigger, about 1,000 times bigger than prokaryotic cells. One of the things you hear most of all with eukaryotic cells is the membrane-bound nucleus. So the nucleus, where the DNA is held, does have a membrane surrounding it. In prokaryotic cells, like bacteria, there is no membrane around the DNA. So we're going to concentrate on the plasma membrane. So let's take a look at the plasma membrane. So the plasma membrane is also called the plasmalemma. So I believe your textbook likes to use the word plasmalemma. Same thing. I usually say plasma membrane. And the plasma membrane, remember the structure of it. So the term used to describe the plasma membrane is the fluid mosaic model, because these molecules that create the membrane are fluid, so they're constantly in motion. It's a mosaic because there are many different kinds of molecules that create the whole membrane. So those molecules are listed for you in bold. The 3 most common molecules are phospholipids. So you can see we have a phospholipid bilayer, and remember that the heads of our phospholipids is going to be hydrophilic, so it faces the water. So the exterior and the interior of the cell are both watery, so they're going to face the outside and inside of the cell. And then we have our internal tails, and the tails are hydrophobic, right? So we have that right here, hydrophilic heads and hydrophobic tails. Well, we also have, right, so that's our phospholipid bilayer. Some of the lipids might have little short carbohydrate groups on it. So I do mention the glycolipids. So you can see that glycolipid term right here. So we can see that the hexagons, this represents our sugar, right? So one monosaccharide looks like a hexagon. We have quite a few of them bound together. So those are little oligosaccharides. Then after our phospholipids, we have proteins. So this is going to be our blue, large molecules that are moving through the membrane. The proteins, they're called transmembrane proteins or integral proteins if they span the entire membrane. So if you look at the word here, right, this is an integral glycoprotein. So it's an integral protein, meaning it goes from the top through, it spans the entire membrane. It goes through the membrane. And then the word glyco tells you that there's some sugar groups attached to it. Then we have peripheral proteins. So peripheral proteins are proteins that are just on one side of of the membrane. So they're either on the inside or outside, but they only take up one side. And I do mention in the 3rd paragraph under proteins that some of these might have oligosaccharides attached to them, in which you call them glycoproteins. Okay, so glyco is for the sugar group. If you have a lipid attached to them, they are called lipoproteins. And I don't see any lipoproteins in this picture you have, but that's what the definition would be. Okay, so let's take a look at the characteristics of a membrane. So the cell's membrane is semi-permeable or selectively permeable. That means that molecules that are on one side of the cell, so I'll just draw a little square here representing one kind of molecule. This molecule may or may not go through the membrane. The membrane is set up to block out molecules it doesn't want to come in and only allow the molecules that it wants in. So one of the tools that the cell membrane uses to allow molecules in is something called a channel protein. So this is an integral protein with a channel, sometimes just called a channel protein. And so it has this space inside of it. So this is cut in half, but you can think of this as like a hollow tube where those molecules can either go out or in. Some channel proteins are gated, meaning that they're closed. You can see how the protein is shut. And so it only opens when there's a certain stimulus that causes this channel protein to open. So it could be electrical charge, or it could be some kind of molecule binding to it. But that's what a gated channel is. So a gated channel is only going to open when there's a certain very specific stimulus to open it. And that's what number 3 talks about in your outline, right? So 3, I have channel proteins, and gated means that it's closed most of the time, but only opens when there's a stimulation. All right, so for number 4, I'm not going to go through this in the outline, but you should review these processes very simply in the book. So you want to review what simple diffusion is, what facilitated diffusion is, osmosis, active transport, endocytosis, and exocytosis. I do have a picture on endocytosis and exocytosis. So this is a kind of, so endo, right, is within. So this is the cell taking something in. So you take in a certain kind of molecule. The difference between endocytosis and the rest of the kinds of transport, diffusion, osmosis, active transport, is that these molecules with endocytosis are really too big to move through the membrane, just through that bilayer of phospholipids or through a channel protein. So they're just too bulky. So what the cell does to take them in is to make a depression within the plasma membrane. So you see the membrane here is actually making a pocket, and it's encompassing this solid particle. And when this pocket completely pinches off, it becomes a vesicle. Okay, sometimes the word vacuole is used. Vacuole is just a larger kind of vesicle, and it brings that substance in within a vesicle or a vacuole. Okay, and there's 3 different kinds of moving these particles in. So let's begin with phagocytosis in your notes. Phagocytosis is engulfing large substances from the outside. It literally means cell eating. So a good example would be a white blood cell, one of your immune cells, gobbling up and destroying a bacteria. So let's pretend that this guy is a bacteria and this is a white blood cell. The white blood cells will literally form a pocket around the bacteria and then pull the bacteria in with a vesicle. Now, the second process would be to digest this bacteria, right? So we have to fuse with a lysosome. We haven't gotten there yet today in this lecture, but the lysosome will contain hydrolytic enzymes and a low pH and acid pH. It'll fuse with that, dump in the enzymes, and then break up that bacteria. So that's the whole process of cell eating. Cell drinking is called pinocytosis. So just thinking about the wine, you have the Pinot Grigio wine or Pinot Noir, but Pinot is cell drinking. So what that means is the cell will constantly take little sips of the fluid, right? So this is fluid around itself. And these little particles are small dissolved nutrients, or they can be solved, whatever it is that's in the fluid. And the cell will just constantly invaginate its plasma membrane. Whatever ends up in that little sip of fluid, so for example, this sip got a little square or diamond, and it will bring it in inside a vesicle. This happened to get nothing, right? So it doesn't matter. The cell will take little sips of the fluid on the outside and just catches whatever it catches in the fluid. So it's a little bit it's not very specific here. It's just drinking whatever's in the fluid. The most specific kind of endocytosis is receptor-mediated endocytosis. So this is a way that the cell can really specifically take in a very specific molecule. I mean, I'm saying the word specific a lot, but this receptor, so for receptor mediated, that means that there is a receptor on the surface of the cell. A receptor is kind of like a baseball mitt. It's a protein that fits only a certain kind of molecule. In this example, it will only fit the star. So these receptors in green only attach to stars. And when there's a star that fits the receptor, it signals for this pit to form, and then it'll drag in just the stars, okay, in a vesicle. So this is a way that cells can control exactly what they bring in. Just for an example, when you bring in sugar and insulin, many of you may have heard of insulin, and you know that insulin controls your blood sugar level. But insulin plus sugar, your glucose, move into your cells with a receptor, okay? So your cells know, once you know that your cells are very aware that they're bringing in this sugar molecule. I shouldn't say aware. Your cells are not aware, but they're as far as we know, each cell is not conscious. But so those are the three kinds of endocytosis. Now let's look at. So this is a picture from your textbook on exocytosis. So this is just the opposite, right? So letter F in your outline. This is the release. So we're moving now, instead of taking things in, we are moving things out. So we have some kind of product inside the cell, some kind of solid inside the cell that needs to be moved out. So these can be wastes that accumulate inside the cell, or they could be something like products that you want to go into the bloodstream, such as hormones. Okay, so secretory products, this word just means it's secreted. So I'm going to put example hormones, right? Hormones are made inside your cell, but they're going to be put outside the cell and they're shipped out through exocytosis. So it's the same process. The cell forms a vesicle and then moves it out, and it fuses with the plasma membrane, and then when it fuses, the contents are released. All right, so the next topic is number 5, extensions of the plasma membrane. This is the microvilli. So the microvilli, as you see here, are these long finger-like, well actually they're short. They're short finger-like projections that come off of the cell, and they are held in place by the cytoskeleton. But their function is to increase the surface area of the cell. So increasing surface area is going to be a recurring concept and a recurring theme throughout your body as we go through this lecture, and all through nature. Nature finds ways, because the cell is so small, it finds ways to pack in a lot of surface area, a lot of tissue, or a lot of structure in a very small space, because it coils or it folds or it pleats or something like that. So this is another way of doing that. So let's just draw one scenario. So this is a cell, a square-shaped cell with no microvilli. And this is a cell with microvilli. Okay, so I'm going to just draw like that. So they take up the same amount of space, right, in terms of how wide they are. They're both the same width. But let's pretend that this cell absorbs substances from this end only. So how much surface do you have for in and out of material? You have that much surface, right? But if you add microvilli, right, to your surface, let's pretend that this was a string, and let's pull it straight, right? So if we pull all that straight, we can see that we have a lot more surface area to move things through, because substances can move not only through the sort of flat part of the membrane, but also through the sides of each microvilli, right? So imagine how much more surface area we have to either absorb substances or to secrete substances. So parts of your body that are very active in absorbing or secreting are going to have cells with microvilli. So one of the places that you might think of automatically that absorbs a lot of things is your small intestine. Because your small intestines, it's evolved to absorb all the nutrients that you have from your food. So it has to be very, very effective and at absorbing that nutrient. So it will be full of microvilli. All right, so I'm going to pause here. So that was just the plasma membrane.